Hello DFAT students! In this video, we'll discuss some important descriptive questions from your last 3 unit tests, water and sewage microbiology. I'll explain each question in detail with diagrams where required, so that you understand both the concepts and their applications. Let's begin. Question 1. Importance of indicator organisms in water microbiology. Why is E. coli considered the best indicator? Answer is, in water microbiology, indicator organisms are microbes whose presence signals possible contamination. They don't necessarily cause disease but point to the presence of pathogens. Coliform bacteria, especially E. coli, are widely used as indicators. E. coli is the best indicator because it originates only from the intestines of humans and warm-blooded animals, survives in water long enough to indicate pollution, but doesn't multiply outside, is easy to culture in the lab, and its presence strongly suggests fecal contamination. Question 2. Describe the principle, medium, and result interpretation of the indole test in the IMVIC series. Answer is, the indole test is part of the IMVIC series used to differentiate Enterobacteriaceae. Principle. Some bacteria have the enzyme tryptophanase, which breaks down the amino acid tryptophan into indole. Medium. The test is performed in tryptone broth or peptone water, which provides tryptophan. The procedure is to inoculate and incubate the culture, then add Kovacs reagent. Indole reacts to form a red compound. Results: a red or pink ring on the surface equals sign positive test, like in Escherichia coli. If no red color, reagent layer remains yellow or brown equals sign negative test, like in Enterobacter or Klebsiella. Thus, the indole test helps identify indole-producing bacteria. Question 3. Differentiate between E. coli and Enterobacter spp based on IMVIC test results. Answer is, the IMVIC test series is a group of 4 biochemical tests used to differentiate enteric bacteria, especially Escherichia coli and Enterobacter species. IMVIC stands for indole, methyl red, Voges Proskauer, and citrate. Indole test detects breakdown of tryptophan to indole using Kovacs reagent. Positive, red ring, for example, E. coli. Negative, no color change, for example, Enterobacter. Methyl red test detects stable acid production from glucose fermentation. Positive, red color, for example, E. coli. Negative, yellow color, for example, Enterobacter Voges. Proskauer test detects neutral end products, mainly acetoin, from glucose fermentation. Positive, red color after adding VP reagents, for example, Enterobacter. Negative, no color change, for example, E. coli citrate utilization test. Checks if bacteria can use citrate as the sole carbon source. Positive, blue growth on Simmons citrate agar, for example, Enterobacter. Negative, no growth green medium, for example, E. coli. Result patterns: E. coli++, indole plus, Mr. plus, VP citrate, Enterobacter, ++, indole, EMSA, VP plus, citrate plus. Thus, the IMVIC series is a simple yet powerful tool to distinguish between coliform bacteria. Question 4. Describe the 3 states of the most probable number, or MPN test. Answer is, the most probable number MPN test estimates coliform count in water. It has 3 stages. First, the presumptive test. You inoculate water into lactose broth with Durham tubes. The gas production is test tube indicates the suspected presence of coliforms. Second, the confirmed test. You transfer positives tubes to selective media, like brilliant green lactose bile broth or EMB agar. Characteristic dark-colored, nucleated colonies with a green metallic sheen. Colonies confirm coliforms. And finally, the completed test. You streak colonies on EMB and check with a gram stain. Gram-negative, lactose fermenters mean confirmed coliforms. Question 5. Discuss the significance of WHO and BIS, standards for potable water in terms of microbial safety. Answer is, the quality of drinking water is directly linked to public health. Both the World Health Organization and the Bureau of Indian Standards have set guidelines to ensure microbial safety of potable water. According to these standards, drinking water must be free from pathogenic microorganisms, especially bacteria like E. coli and coliforms, which indicate fecal contamination. The permissible limit for E. coli and thermotolerant coliforms is 0 per 100 ml of water. Total coliforms are also not acceptable in treated drinking water supplies. Significance: these standards protect people from waterborne diseases such as cholera, typhoid, dysentery, and hepatitis. They guide water treatment plants to maintain safe chlorination, filtration, and monitoring. They provide a uniform benchmark for assessing water safety globally, WHO, and nationally, BIS for India. In summary, WHO and BIS standards are vital because they ensure that water is safe, pathogen-free, and suitable for human consumption, thereby preventing outbreaks and safeguarding community health. Question 6. Compare qualitative versus quantitative microbiological tests for water quality. Give one example each. Answer is, we have two main methods here. First, we have qualitative test. The purpose is to detect the presence or absence of indicator organisms in water. Principle is to detect even a small number of pathogens. The test focuses on detection rather than counting number. Example is presence or absence test for coliforms, which gives a color change indicating contamination. Second, we have quantitative test. The purpose is to estimate the actual number of microorganisms present in a given volume of water. Principle is to provide actual microbial load. It is useful for comparing with standards and assessing treatment efficiency. Example is the most probable number, MPN test, which gives statistical estimate of coliforms per 100 ml of water. Summary. Qualitative tests answers if the contamination is present or not, while quantitative tests answers how much actual contamination is present in terms of count. Question 7. Write a short note on the public health implications of detecting E. coli in drinking water. Answer is, presence of Escherichia coli in drinking water indicates recent contamination with human or animal feces. Fecal matter may also carry harmful microbes such as Salmonella, Shigella, Vibrio cholera, hepatitis viruses, and protozoa. Even a small number of E. coli suggests that water is unsafe and may cause outbreaks of diarrhea, cholera, typhoid, dysentery, and hepatitis. WHO and BIS standards organizations mandate 0 E. coli per 100 ml of potable water. Detection means the water supply fails safety standards. Immediate measures include tracing contamination sources, improving chlorination filtration, and issuing boil water advisories. Question 8. Describe the microbiological processes involved in a septic tank, highlighting the roles of hydrolytic, fermentative, and methanogenic microorganisms. Answer is, in a septic tank, waste is broken down by different groups of microorganisms under anaerobic conditions. First, hydrolytic bacteria break complex compounds like proteins, fats, and cellulose into simple molecules. Next, fermentative or acidogenic bacteria convert these into volatile fatty acids, alcohols, hydrogen, and carbon dioxide. Finally, methanogenic archaea transform these products into methane and carbon dioxide, stabilizing the sludge and producing biogas. All these microbial groups work together to stabilize the sludge in the tank. Thus, septic tanks work through a sequence of hydrolysis, fermentation, and methanogenesis. Raw sewage, it has a very high microbial load, including a lot of pathogens. After going through septic treatment, the microbial count does reduce, but the effluent may still contain coliforms. That's why, safe disposal or sometimes even further treatment is really needed. Question 10. Outline the advantages and limitations of septic tanks as decentralized sewage treatment systems from a microbiological perspective. Septic tank advantages include 1. Septic tanks use natural microbial processes, hydrolytic, fermentative, methanogenic, to break down waste. 2. They reduce organic load, pathogens, and odors effectively at household or community scale. 3. Produce biogas, methane plus CO2, that can be used as an energy source. 4. Simple, low cost, and do not require advanced infrastructure. There are also some limitations include 1. Incomplete treatment. Effluent still contains pathogens and nutrients, needs further polishing. 2. Sensitive to overloading, toxic chemicals and antibiotics, which can disrupt microbial activity. 3. Sludge accumulation requires periodic desludging. 4. Methanogens are slow-growing, so treatment efficiency may fluctuate. Summary. Septic tanks are a cost-effective decentralized system, but from a microbiological perspective, they provide partial treatment, and thus the effluent may still cause pollution. Therefore, further treatment processes to ensure safe disposal. Question 11. 1. Explain the importance of microbes in water and their role in public health. Microbes recycle nutrients and maintain ecosystems, which is really important. Answer is, microbes are natural inhabitants of water and play an important role in the ecosystem. Beneficial microbes help in self-purification of water, nutrient cycling, and maintaining ecological balance. However, the presence of pathogenic microbes, such as E. coli, Vibrio cholerae, Salmonella, and viruses indicates fecal contamination. These can cause serious waterborne diseases like cholera, typhoid, dysentery, hepatitis, and gastroenteritis. Therefore, regular microbiological monitoring of water is essential. Standards set by WHO and BIS ensure that potable water is free from coliforms and pathogens, safeguarding public health. Question 12. Explain the steps involved in municipal water treatment plant. Answer is, municipal water treatment ensures that raw water is made safe for drinking by removing suspended solids, pathogens, and harmful chemicals. The main steps are screening. Large debris like leaves and plastics are removed. Coagulation and flocculation. Chemicals such as alum are added to clump fine particles into larger flocks. Sedimentation flocks settle down, separating solids from water. Filtration. Water passes through sand, gravel, and activated carbon filters to remove smaller particles and microbes. Disinfection, chlorination, ozonation, or UV treatment kills pathogenic microorganisms. Storage and distribution. Treated water is stored in reservoirs and supplied through pipelines. Question 13. Write short notes on any two methods of water treatment other than reverse osmosis or RO. Besides reverse osmosis, there are other methods of water treatment include UV treatment, which, kills microbes. Other is chlorination is a really economical disinfection method, while ozonation is a powerful oxidizing treatment. 1. UV treatment. In ultraviolet disinfection, water is passed under UV light. UV rays damage microbial DNA, making them unable to reproduce. It is fast, chemical-free, and effective against bacteria, viruses, and protozoa. 1. Chlorination. Chlorination is the most common method of disinfecting water. Chlorine or bleaching powder is added to kill pathogenic bacteria, viruses, and protozoa. It provides a residual disinfectant effect that protects water during storage and distribution. In conclusion, I hope I could answer all the questions nicely and in simple language. Thank you for watching, and don't forget to like, share, and subscribe for more lessons.